Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base Biotech podcast, and this is number 43, and it's going out on January the 23rd. My voice is getting back to normal, whatever normal is, and it seems everyone around here has had the same illness and also struggled for weeks with it. Lots of people sent me Northern Lights photos this week. Seems like it's been visible everywhere in the Northern Hemisphere, except here, where it's been raining, and when it hasn't been raining, it's looked like it's going to. Nice to see all the photos though, although I could do without the first sentence in all of those texts being, "Did you see the aurora?" No, I didn't. Last week we had 3 interviews and 5 guests. It was definitely a bumper episode. We chatted about treating blood cancers with Nishit Khandelwal, co-founder and CEO of Artiva Therapeutics, Ulrich Seiten, CEO, and Katerina Cantel, founder and CSO of Adalion, and Craig Ritchie, founder and CEO of Scottish Brain Sciences, and Deborah O'Neill, CEO and CSO of Nova Biotics, as well as chair of the Life Sciences Board of Opportunity Northeast, which runs One Biohub. This week we have a conversation with Lisa Guerrettaz, Executive Director, Pharmacology and Translational Science at Artiva Biotherapeutics, and we also chat with Avacta Therapeutics CEO, Christina Coughlin. If you do have any news or suggestions for who should be on the podcast, or indeed any other suggestions or comments, please do get in touch at jim@deeptechdigest.com. which is also the address you can use if you're interested in advertising with us or sponsoring a podcast episode, or indeed for event coverage. As my voice is improving a little bit, let's bring back our weekly look at the news you may have missed over at deeptechdigest.com, and fortunately there was a lot of it this week. Fast Track designation has been given to a treatment for geographic atrophy secondary to AMD. There's a new way to disarm antibiotic resistance bacteria and restore healing in chronic wounds. And a first-in-human drug trial has been shown to lower high blood fats. An AI framework has been launched to improve life sciences decision-making. AirNexus Therapeutics, which focuses on COPD, has launched, and the University of Pennsylvania BioNTech and OUP have launched a $50 million life sciences fund. The Novo Nordisk Foundation has granted $860 million to boost innovation in Denmark and Europe. I wonder if we'll see a hub emerge in Greenland. AbbVie has announced top-line results for its Phase 3 lymphoma trial. There have been positive results from an insomnia trial, and a treatment for Wiskott-Aldrich syndrome has been approved. Scientists have uncovered hidden cells fueling brain cancer and a drug that could stop it. Brainomics' E-Lung AI imaging technology has been selected for a Boehringer Ingelheim pulmonary fibrosis trial, and you can read all of these and plenty more at deeptechdigest.com. Alright, now that we're somewhat back on track, let's get to the first of this week's interviews. Artiva Biotherapeutics is a clinical stage cell therapy company developing off-the-shelf natural killer cell therapies for autoimmune diseases and cancer. To tell us about the company and how its work is making a difference is Artiva Executive Director, Pharmacology and Translational Science, Lisa Guerrettaz. So maybe then to get started, if you could give me a bit of background on the company and the history of Artiva.
Thanks, Jim. I'm excited to share a little bit about Artiva today and what makes us unique. We're a clinical stage biotech company dedicated to advancing NK cell therapies or natural killer cells for autoimmune diseases. So Artiva originated as a spinout from GC Cell. This was a pioneering Korean company that established the NK cell manufacturing process. And this was a little over a decade ago. So leveraging that foundation, we've developed a highly scalable platform for producing these NK cells. And one thing that I should note that's important is that we hold exclusive rights outside of Asia Pacific for all the programs, but including our lead candidate, AlloNK.
So maybe if you could explain what AlloNK is and how it works.
Sure, I'd love to. So, AlloNK is Artiva's off the shelf NK cell therapy designed to enhance ADCC, also known as antibody dependent cellular cytotoxicity. But before I jump into AlloNK and how it works, it might be good to provide a little context on Artiva's approach. Back in 2022, there was seminal work done at an academic center in Germany led by Georg Schett. This work showed that patients with autoimmune diseases who had received CD19 autologous CAR T cell therapy were able to achieve deep B cell depletion. And that's what led to durable clinical responses and remission. So these are patients who had failed multiple targeted therapies or were on chronic immunosuppression. And remarkably, these patients went into remission and they were able to come off of all their background medications, which was pretty phenomenal and transformative for the field at the time. So essentially, where Artiva comes in, we learned from this auto CAR T experience that achieving deep B cell depletion, followed by reconstitution of the immune system with naive and transitional B cells, this could truly change the game for autoimmune diseases. You know, the only problem is auto CAR T therapy is quite complex. It requires individualized manufacturing, so taking the patient's own cells. It requires specialized centers. And it often comes with safety challenges that need to be managed very closely in a hospital setting. So these include high rates of cytokine release syndrome, also known as CRS, and there are other neurological toxicities that make this a challenging approach to scale to large chronic autoimmune populations. So where does Artiva come in? So we asked ourselves, can we deliver the same type of efficacy and deep B cell depletion that drives these clinical responses in autoimmune patients, but is also a therapy that's accessible to rheumatologists and can be scaled to treat a large number of patients? And so AlloNK is our answer to that. As I mentioned earlier, it's an off-the-shelf NK cell therapy. It's designed to work alongside monoclonal antibodies, so in this case, anti-CD20, such as rituximab or obinutuzumab. And the idea is to deliver consistent and complete B cell depletion. So another benefit of AlloNK is that it can be administered outpatient. So it only requires about 5 to 6 visits to an infusion center, which is different than CAR T cell therapy and T cell engagers, which is another modality in this space. So I should also mention, AlloNK is highly scalable. Artiva is able to manufacture thousands of vials of AlloNK with 1 billion cells from each cord blood unit. This allows physicians to deliver a standardized, off-the-shelf, cryopreserved product. It's thawed the day of treatment and infused IV within 5 to 10 minutes. So this is great for rheumatologists. It's more similar to IV monoclonal antibody administration. Rheumatologists have been delivering that for decades. So this really fits in line with their comfort level. And overall, I should say that Artiva's goal is to bring patients the same durable clinical benefits associated with auto CAR T, but in a way that's more practical and delivered through a regimen that's tolerable, scalable, and easily integrated into everyday community rheumatology practices.
You mentioned deep B cell depletion a couple of times. Could you explain what that is?
Sure, happy to do so. So maybe it's important to start with why B cells matter. So one can think about B cells as the body's security guards. They're supposed to protect you from intruders like viruses and bacteria. And unfortunately, in autoimmune diseases like RA, B cells become pathogenic. So instead of protecting you from infections, a subset of them start producing inflammatory signals. They present self-antigens and begin activating T cells, which essentially leads to fueling the immune system to attack your own tissues. So the idea behind B cell depletion is it's the process of clearing out all the circulating and tissue B cells from the immune system. This would include wiping out the pathogenic B cells. The goal really is to eradicate pathogenic B cells that keep the disease going rather than just suppressing them like some other modalities may do. So anti-CD20 antibodies like rituximab, they do this to some extent, although the depth and consistency of depletion is inconsistent. So that leads to a limited clinical response or durability in some patients. You can imagine if there are any residual pathogenic B cells around, they'll still allow the disease to persist. So it's really important to get as many of them depleted as possible. As I said, many patients receive B cell depleting antibodies today, but it's just inconsistent. It could be many reasons why, but we believe that with AlloNK's mechanism of action, which is enhancing ADCC or the ability to target these B cells with antibodies and kill them more efficiently, this is the way to move forward. So I can explain a little bit how that works. So first, in our setting, you have the antibodies administered first. These antibodies go in, such as rituximab. They go in, they coat B cells, and then AlloNK would be administered. It comes in, it sees the targeted or coated B cells, and then it kills those more efficiently. So by using AlloNK as an ADCC enhancer, the goal is to achieve uniform and consistent depletion of B cells for a period of time. When you do this, what happens is the bone marrow starts generating new B cells, naive and transitional. So you're essentially reconstituting your immune system with newly generated B cells.
And so, what led to you choosing refractory rheumatoid arthritis as your lead indication?
So, refractory RA describes a population of patients whose disease activity remains high despite any prior treatments with multiple biologics or targeted therapies. These patients face ongoing pain, fatigue, and functional limitations because there just aren't great options out there to sufficiently control their disease. In the US alone, it's estimated about 150 to 200,000 patients are facing RA without any option. So there's clearly a defined unmet need. What we do know about RA, it's a well-defined patient population. There's established clinical endpoints, and currently rituximab is already approved in RA. So it makes sense for us to go into combination with rituximab. So given AlloNK's tolerability profile that we've seen so far, its scalability and its outpatient friendly regimen, we believe it's well positioned to address this unmet need in RA and to deliver a therapy that's able to lead to durable clinical responses in all patients.
And so, what are the advantages of AlloNK compared to CAR T?
Yeah, so CAR T and T cell engagers, those are both T cell directed therapies. They can be very potent, but the problem is they tend to come with higher rates of those safety risks, such as cytokine release syndrome and neurotoxicity that I mentioned before. So that high risk, it makes patients and physicians for that matter nervous about using it. It also requires specialized centers and intensive monitoring by physicians. So AlloNK from the outset was designed for outpatient use. It's a defined course of treatment. There's no need for specialized infrastructure. And importantly, there's no expectation of long-term persistence of the cells. And what we're learning from auto CAR T is actually in autoimmune disease, you don't really need long-term persistence of the cells the way that you do in oncology. So one fact about NK cells is they don't actually, when they're non-engineered, they don't stick around a long time. There's no need for long-term proliferation or persistence in the body. And in clinical trials to date, they haven't been associated with severe CRS, neurological toxicities, or graft versus host disease, which is often seen with T cell directed therapies. This regimen with AlloNK, it's delivered in standard infusion centers. So patients get to go home after treatment. It fits pretty nicely within current rheumatology workflows rather than being at a specialized cell therapy center. So at the end of the day, it's just more accessible for patients.
Could you tell me a bit more about the FDA fast track designation that you got and about the clinical trials that you're doing?
So the FDA granted fast track designation to AlloNK in combination with rituximab for refractory RA, reflecting both the unmet need I mentioned and the potential of deep B cell depletion as a new treatment approach. So currently, Artiva has a phase 2 basket trial. This consists of rheumatoid arthritis, Sjogren's disease, idiopathic inflammatory myopathies, and systemic sclerosis. Additionally, we have a company sponsored trial in SLE and lupus nephritis, and we have an investigator initiated trial in a community rheumatology practice. So, across all of these studies, AlloNK is dosed at 1 billion or 4 billion cells with rituximab or obinutuzumab. And importantly, there's no required hospitalization or patient stagger in any of these trials. So the regimen involves 6 outpatient visits over about 20 days with Cy/Flu conditioning, followed by 3 AlloNK infusions and 2 rituximab doses that are currently given in the way that's on the label for rituximab dosing.
And you recently announced some results. Could you tell me about those?
Sure. Artiva in the November webcast shared initial safety data in 32 patients with autoimmune diseases, specifically RA, SLE lupus nephritis, Sjogren's disease, and systemic sclerosis as of October 1st, 2025 data cutoff. As of that data cutoff, there was no CRS, ICANS, or GVHD, which are all cell therapy related toxicities of interest. There was also no AlloNK related serious adverse events. So overall, as of the data cutoff, the tolerability profile was consistent with rituximab, obinutuzumab, and Cy/Flu. And importantly, the translational data demonstrated complete B cell depletion, and this was using a high sensitivity assay in all patients evaluated. Importantly, I wanted to just mention the high sensitivity assay. Not everybody uses one of those, but the monoclonal antibody field does. And they do that because they want to make sure that they're really getting that good deep B cell depletion. And standard assays don't necessarily allow you to detect any residual B cells, but we employ a high sensitivity assay. And using that assay, we see a complete depletion of B cells. We do expect the B cells to come back, as I mentioned before. And so they reconstitute with predominantly naive and transitional B cells. So to date, from our November disclosure, it looks like we're moving in the right direction for achieving CAR T-like B cell depletion, but with a simpler NK cell plus monoclonal antibody approach.
Are there any other potential uses for AlloNK?
Sure. So, as I mentioned, we're looking in Sjogren's disease, lupus nephritis, SLE, systemic sclerosis, and the myopathies. We're also looking at a couple others in an investigated initiated trial. Really, this approach can be relevant for any B cell dependent disease and anywhere where anti CD20 antibodies are already part of care or being explored. But the good news is that it can be combined with other targeted antibodies. So, for example, CD19 or CD38, those antibodies might allow you to reach different subsets of B cells that might be important. So, yes, I think AlloNK, because it's not an engineered cell with a specific target, it has more flexibility in that it can be combined with different monoclonal antibodies or innate engagers.
Do you have any partnerships currently? And what's investor interest like in NK cell therapies compared to CAR T or even bispecifics?
I think there's a growing interest in approaches that can deliver the power of cell therapy, but in a practical, scalable way for autoimmune disease. And NK cell platforms are very much part of that conversation, given their great safety profile. We're consistently engaging with clinicians, collaborators, academic institutes, and potential industry partners who see the need for a safer, outpatient friendly option that can still deliver the durable clinical responses that we're hoping for. So, investors, they often ask 2 questions. Can you really deliver deep B cell depletion safely in an outpatient setting? And can you manufacture at scale at a reasonable cost? And I'd say Artiva's data and manufacturing background are aimed directly at those points. So, I think we're in a good place to move forward.
Do you think that AlloNK will be more viable in some places where CAR T infrastructure is limited?
I'd say to date, our experience in autoimmune disease has shown the regimen can and has been able to be used in community rheumatology practices without the specialized oncology oversight or the inpatient hospital monitoring that's required for CAR T and T cell engagers. We use existing infusion center infrastructure similar to rituximab or other IV administered monoclonal antibodies, and patients get to go home after their visits. So that design was intended to support the broad adoption in settings where CAR T centers are not readily accessible.
What do you see as the future for AlloNK?
Sure. In the near term, we're focused on generating clinical response data in RA in 2026 and aligning with regulators on a pivotal trial design in refractory RA. In parallel, we're using our ongoing trial to identify additional autoimmune indications to pursue for AlloNK in development. I'd say longer term, the vision is an outpatient immunotherapy platform that works alongside antibodies to provide durable disease control without chronic broad immunosuppression. We're really excited about the potential of AlloNK to break away from the rest of the field by advancing the first deep B cell depleting therapy into a pivotal trial in refractory RA. At the end of the day, Artiva's vision is to bring cell therapy activity with a biologics-like accessibility and tolerability. Going from the complexities of CAR T cells to the simplicity of an NK cell that's not engineered. I think the future is really exciting.
We've talked about natural killer cells before, and it was an interesting subject. Certainly looks like AlloNK is set to make a big difference in a lot of different areas. And it was also nice to chat about San Diego as well before the interview, one of my favourite cities. The first time I went there was just after we moved to Nevada. It was in the middle of January, and the day that we left Canada it was -15, with a wind chill quite a bit lower than that. So on a quick trip to San Diego, it was 19 degrees or 66 Fahrenheit, and everyone we chatted with was apologising for the chilly weather. Alright, let's move on to Avacta Therapeutics, a clinical stage biopharma company developing Precision, a tumour-activated oncology delivery platform. The company recently announced FDA clearance of the investigational new drug application for the first precision peptide drug conjugate. To tell us about the company's work is Christina Coughlin, Avacta's CEO. You've had quite an interesting career to date, so I wonder if maybe you could give me a bit of background on that.
Sure. Thanks, Jim. So I'm an oncologist and an immunologist. What that means is that I was able to get both my medical degree as well as PhD in science. I get asked a lot, why did I go into medicine and why did I go into oncology? When I was young, late teens, early 20s, I lost 3 important men in my life. My grandfather, my godfather, and my best friend's father all died of cancer. And so as a very young student, I had almost a vendetta against this disease. And so I kind of always knew, I used to, be the doctor to my stuffed animals when I was a kid. I always wanted to be a doctor, but oncology was really a very personal decision for me. And immunology was because at the time we were just figuring out in the late 90s that the immune system really plays a deep role in the development and in the treatment of cancer. But I will say getting both the scientific training as well as the medical training, I believe has led to some of my success in industry. There's something really special about going through scientific training as well, true rigorous scientific training postdoc that really teaches you to put a scientific program together. And I think that has been, I've relied on that quite a bit in my career, in addition to really treatment of patients and treatment of patients with cancer, understanding what those patients have gone through in terms of, some of our therapies are really quite toxic. And having done that for a number of years, seeing patients go through that is what really brought me, honestly, to Avacta and why I'm just in love with the precision technology.
Is that a normal pathway for people to have both of those backgrounds?
It's not abnormal. It's not easy. It takes quite some time. The time that it takes to get both an MD and a PhD is anywhere from 7 to 9 to 10 years. And so it's quite a bit of training. This is also in the United States, it's also after one does university. So it's 4 years of university, 7 to 9 years to get both doctorate degrees. And then one is ready for medical training, which is residency and fellowship. So it takes quite a bit of time. It's not an easy decision to make when all of your friends are already having jobs and out, and you're still studying and still in school. I will tell you a funny story. My mom just kept asking me, the different transitions. Christina, is this a job? Christina, do you have a job? And I would have, no, I'm in training again, Mom. So, she goes to the fancy graduation at Penn. Now you're going to get a job. Well, no, now I have to go into medical residency. Oh, how long is that? 3 years. Okay, finish residency. Are you going to get a job now, Christina? No, Mom, I'm going to do a fellowship. So it's quite a commitment, but I will say very good training at Penn.
For sure, yeah. I thought you were going to say that you were telling her that you were going to try and aim for getting through that and then going straight for retirement. But yeah.
I did scare her at one point and said, you know what, I think I might get a master's in statistics. Oh, Christina Marie. And I know I'm in trouble when my middle name comes out.
From your own background, I wonder then if you could tell me about Avacta.
I've been with Avacta now for over 3 years. I started as a director and was hired by our previous chairman, Elliot Forster, to come in and to help with clinical development. At the time, we were struggling a bit with the first drug, AVA6000, now known as faridoxorubicin, to get that trial enrolled. And it was as the data for that started to roll out that I was feeling like I really wanted a permanent role in the company. And it was really about just how unique the platform is and how it speaks to something that I experienced early in my career, which was as I was treating patients and treating a lot of the toxicities, noting that the drugs that we use in cancer generally shrink tumors. That's why we use them. That's why we put patients through those toxic drugs. But isn't there a way that we could somehow target these drugs directly where we need them to go, which is the tumor? And that's exactly what Precision does. So Precision is a very simple platform. It's a small little peptide. It's small enough to not be noticed by the immune system, which is a key advantage. But it's a peptide that when we attach it to a given drug, it inactivates that drug. And so why is that important? Well, the peptide then is specifically removed by an enzyme that only exists in the tumor. So the tumor, thinking that it's metastasizing, which is what this protein FAP does, it's actually feeding itself that toxic drug. But the peptide has an extra benefit. We call it mask and release. And so it masks the toxic drug from normal tissues. Normal tissues cannot remove the peptide. And so it's a very simple idea, and it's almost elegant in how simple it is. And when you look at the pipeline and what our scientists have been able to do with this peptide by modifying both ends of it, we've really been able to create some novel medicines that are just about to hit the clinic. So our first medicine, faridoxorubicin, has been through its phase 1 testing. We now know that we can concentrate the drug right in the tumor microenvironment. We dramatically reduce the toxicities. It does everything that we planned for the platform to do. The next benefit is really to do a better job of keeping it out of the bloodstream, away from the normal tissues, but also to create this sustained release mechanism. So hold the drug right in the tumor. So the enzyme now not only cleaves it, but it holds onto it, cleaves it slowly. And so the drug gets released over time right in the tumor where it should be. It's a fascinating mechanism and really simple. And so now what we do is take that new IP and implement it across a number of different drugs.
Could you tell me a little bit more about dual payload drugs and how you kind of mentioned a little bit about how they work, but why they're becoming popular? Is it that targeted approach?
So let's go way back again to my original training in oncology. We rarely give a single anti-cancer drug alone. Generally speaking, in oncology, we treat with we call it a cocktail or groups of drugs, combinations of drugs. One of the things that our scientists were able to invent on this peptide, we can put a linker, which is just a small little chemical group that goes between the peptide and the drug. And what that allowed us to do is insert another binding position. And so now the peptide can bind not only to a single cancer drug, it can bind to two anti-cancer drugs. Why is that important, you should be asking me? Well, if we can target the tumor, give an anti-cancer drug, but also at the same time give another drug that hits a resistance mechanism, we can now prevent that inevitable fact, which is cancer tumors are very good with their ability to resist our various mechanisms, which is why ultimately we treat with combinations. And so what we can create now, instead of giving two different drugs into a vein and hoping that they both get to the tumor in any kind of concentration that's effective, we can now have them both released by this precision mechanism, concentrate both drugs right in the tumor microenvironment, right where they should be, and protect normal tissues. We think that this is going to create highly effective combinations by targeting the tumor and the resistance mechanism in parallel, but with a single medicine that also protects normal tissues from the toxicities. The toxicities is what we struggle with in combination therapy, because we have to figure out exactly how much of each of these toxic drugs we can give at any given time, kill the tumor, but not kill the patient.
And is the delivery method the same, or are you targeting the actual tumor?
The delivery mechanism is the same. And that one little chemical group, we call it a cap, that we add, the tumor is able to hold on to it. It's the FAP enzyme that is able to hold on to the drug, keep the active or the joined up drug, the conjugated drug right in the tumor, and then slowly release both active payloads. It's a fascinating mechanism. We're really looking forward to getting the first sustained release into the clinic. That one will start in March of next year. We're aiming for the first quarter for the first patient with that one. The dual payload will take us a little bit longer to get to the clinic. We just published in October some early data showing that we can get both drugs to release. They're both intact. And we've done it with a number of different combinations, I would say. And so we're in the process now of selecting which of those will go first, which will go second, and moving those forward in parallel with that first sustained release mechanism of exatecan, which will hit the clinic in the first quarter.
Does AI modeling help with that process of knowing how to do this?
That's a great question. We were just featured at London Life Sciences Week as the company to watch in using AI. And so we use computer modeling of the FAP active site. Jim, I'm going to go nerd for you on just a minute here, but we use a computer model of the FAP active site and can now position all these different chemical groups and understand from the computer. The computer can search through a number of different chemical groups and predict for us based on in vitro data that we use to load into the engine, can help us to predict what these medicines with the different caps and linkers, what they'll look like when they come out. The other way that we've used AI is to make our trials much faster and much smarter. So there is a database with a company known as Tempus. It's 200,000 human tumors that they have profiled at every biologic level, from DNA to RNA to protein, understand truly deep biology of these tumors, but they also have all of the clinical data associated with those patients. So it's a fascinating data set. 200,000 patients, 7 million data points. And we've leveraged that database and asked the data scientists there a number of different questions. Which patients would be most susceptible to a FAP exatecan? What are the resistance mechanisms that we would see to a topo-1? And so it's a different way to use AI in drug development, but what it's really doing for us is driving smarter and really faster trials. Now we understand who are the exact patients. What is the biology of the exact patients that we'd like to go into? What are the highest likelihood of success places that we can take drugs? And that's exactly how we designed the clinical trial for the next medicine that'll start in the first quarter. We call it FAP exatecan, or AVA6103. And that one was both designed using those AI modeling techniques, but the trial has been driven by those AI techniques as well. imagine trying to explain that to an audience in 5 minutes, but it was fun. It was fun, and it sparked a number of great conversations at the Life Sciences Week.
You were mentioning how this can tell you who or the patients that this will be successful for. Do you go beyond that to find out why it's successful in some people and not others in order to future-proof what you're doing to be able to develop better drugs?
It's a great question. And this is where the patients and the investigators come in. A part of phase 1 trials in oncology that can be very challenging to accomplish is a biopsy after the drug is administered. Imagine on this trial, and we were pretty successful in our first drug, faridoxorubicin. It's optional for patients, but generally speaking, the oncologist treating them explains this biopsy, going through this procedure 24 hours after you've just gotten the first dose of this experimental drug, it's a hard decision to make. And it's a very selfless decision that these patients make, because that biopsy isn't going to, it's highly unlikely that it will benefit the individual patient. What it does is it helps teach us a lot about how the drug is distributed, how it gets into the tumor, how quickly it gets into the tumor, how much is being released. We study every aspect of those biopsies, and you can see even new biopsy data that will come out in our next data release, which will be later this month. The other biopsy that we take is at the time of resistance, or at the time that the tumor grows too much for the patient to continue on the drug. Those biopsies are like gold to a company like ours, trying to understand what happened, what was that resistance mechanism. And that's some of the benefit that we have in the Tempus database, is that we've learned through in vitro and laboratory studies what some of these resistance mechanisms are. We can then apply them to the database and understand what patients, what diseases, where are we seeing those resistance mechanisms. And so I think as a field, oncology is really becoming very, very data heavy. We're starting to really learn how to use data from patients who are even just treated with standard of care therapies. And so using those big data to help us to better design, I would say, smarter drugs is exactly where Avacta is. And I think Avacta is really out on the forefront of this and really leveraging some of these big data sets, Cancer Genome Atlas, Tempus. There's a number of them out there. The UK, through the NHS, has a number of databases, and we've looked at all of them, I'll be honest with you.
Clearly, that's one of the challenges, but are there other challenges with developing systems like this?
Some of the challenges that we face is in that clinical trial mechanism. It does take time. It takes time to enroll. It takes time to slowly escalate. But it's important time that we spend. It's important to pay attention carefully to the safety of all of the patients as we escalate. What Avacta does, most companies will look at these similar mechanisms, is that the science grows up at the same time that the clinical trial. It's almost a revolving door of data. And so the clinical trial data that we generate, it's on individual patients, and we feed that right back into the discovery engine. And what you can see in many of the presentations that the company has given, we learned so much from that first drug, and especially from the generous patients that enrolled in the clinical trial, the biopsies that were taken. That trial did take us quite a bit of time, but we learned so much, and that fed right into the exatecan program as it was growing up. So probably one of the biggest challenges that we have is time. It does take time for each of them, but it's important to gain as much understanding and knowledge and feed that back from the trial, and then feed that right back into the drug development engine.
Right, because I guess everybody looks when people publish results and publish clinical trial data that they're looking at efficacy, but there's also so much more to it than just did it work. It's how did it work? Why did it work? Why did it not work? There are so many questions that you learn from for future use.
You hit the nail on the head. And a company like ours, a platform company, anything that we can do in those trials, anything that we can learn that feeds right back into the discovery engine, you're 100% correct. People do focus on the efficacy. As the CEO, as someone who's taking care of that entire pipeline, I almost focus just as much on the translational data. What is going to help our pipeline? What makes it that next drug better. That's exactly where the sustained release mechanism came from. We observed with that first drug that it was all cleaved very quickly. And so the peptide drug conjugate disappeared almost too quickly. It wasn't being held in the tumor. And so fixing both of those or inventing new chemical groups that allowed us to hold the drug in the tumor, release it more slowly, that's exactly how we could take such a potent drug like exatecan forward. We're really proud of that development.
What are the advantages over traditional antibody drug conjugate delivery?
Great question. And we've benefited from all of the data that's been collected on the antibody drug conjugate, the ADC drugs that are out there. We think there's a couple of key advantages here. The first is FAP as a target, it's kind of the target of our medicines. It's expressed in 90% of solid tumors. And so it has a very, very broad market as opposed to an ADC, which is limited by the target of the antibody. So, for example, one of the most successful ones out there in HER2 is limited to HER2 positive cancers, whereas we can go with this platform into virtually every solid tumor. The second is it doesn't include a biologic element. Our peptide is small enough that in terms of manufacturing, it's a small molecule. So we don't need a cell-based system to manufacture a part of the drug. We make the small little peptide. It's a chemical, essentially, and we attach it to the drug. Very simple and very cheap manufacturing process. But the third, along with that bystander effect, so it's released outside of the cell. It can impact then any cell around it, these drugs. But because it doesn't have that bulky biologic antibody, it also has great tumor penetration. So, for example, with that second drug that I mentioned, our FAP exatecan, we see that being held in the tumor biopsy within minutes. And so it has outstanding, great tumor penetration, goes right into the tumor because it binds to FAP and sits right in the active site. So we think that there's a couple of key advantages: manufacturing, price of that tumor penetration, but also the broad nature of FAP expression speaks to a very large market opportunity.
And what kind of cancers is this going to be applicable to?
So each individual drug, we think about what the payload or the drug that we attach the peptide to, where that payload is applicable. Because FAP is expressed in every solid tumor, and by solid tumors, I exclude what we refer to as liquid or hematologic. So leukemias and lymphomas won't have any place with this platform, but every other cancer, from brain cancer, head and neck cancers, salivary gland, breast cancer, pancreatic, if there's an organ with a cancer, we can address it. And so we're truly excited about that, given that it's a platform, though, and these different cancers respond to different drugs. We are looking to develop an entire suite or pipeline of precision medicines to address all of those different diseases, but using that same peptide with its cap and its linker.
The different kinds of cancers, what are you looking for the end result for patients? Is it to kind of stop it in its tracks and prolong life, or is it to completely get rid of the tumors?
We'd like to do both. I always say, in my clinical practice, we do the scans and we see how the tumor is behaving. The question, though, that patients ask, they never ask, hey, doc, what % decrease did you see in my tumor? They ask, hey, doc, how much time do I have? And so the ultimate goal for the oncologist is to extend survival. In the setting of metastatic disease, so diseases, cancers that have spread beyond that initial organ, we look for survival. As oncologists, we use some what we call surrogate endpoints, meaning what is the response rate, what is the progression-free survival, but ultimately the goal is to extend the patient's life, but also it's to make these therapies a lot more friendly to patients. The goal here is to not only create that extended to survival, but it's also to provide, we call it hope without compromise. So not compromising day-to-day life, feeling good, feeling better. What I love about the precision technology is that it really has shown to dramatically reduce the toxicities that patients endure. And because we've reduced those toxicities, we're able now to treat patients for longer with a given mechanism, all feeding into that important endpoint, which is survival.
And you recently presented some preclinical data. Could you tell me about that and also how the presentation went down?
So we presented a whole suite of the preclinical data of exactly how the dual payload works. We presented the rationale. We get asked a lot, why not just combine your drugs with this given drug? Well, we like the idea of concentrating both drugs at the same time in the tumor instead of allowing the bloodstream to deliver each individual drug. We like the idea of treating with a potent anti-cancer agent like a topo-1 inhibitor and delivering a drug that can overcome a resistance mechanism at the same time. And so it was a bit of a deep dive into exactly how we've been able to make this work. It's important to note the first dual payload ADCs, antibody drug conjugates, are just hitting the clinic now. And, back to your question about what are the advantages, we think that there are some key advantages here. And those were some of the exact conversations that we had surrounding that presentation at the Molecular Targets meeting. It's pretty exciting days, and we had a lot of oncologists drop by with their own ideas of what the combinations should be. So we are knee-deep in putting together our first ideas of what the combinations should be. The first ideas going forward is that, broadly applicable, that topoisomerase 1 inhibitor. There's multiple different versions of that, but putting that together with drugs that would inhibit the resistance mechanism. We're truly excited about this and really looking forward to moving that one in the clinic quickly.
Is it generating any interest from big pharma companies in either licensing or partnering?
So I can't disclose too much of that here, but I would say that we're having a number of very interesting combinations. All of the pharmas out there have their own toxic drugs. We've been trying in the field of oncology to attack resistance mechanisms for quite some time. So yeah, it's going to be an interesting, I'm giving it about 6 months for us to figure out exactly what are those first few combinations that we're going to take forward. It's a great question.
Right. And so what does the clinical trial pathway look like from this point?
We have actually shown that the mechanism works through all of our in vitro work, and that's our 2-dimensional cultures as well as 3-dimensional, tumors that we actually grow in a test tube. The next step is to show efficacy, but also that tumor to plasma, really understanding the mechanism here. It's like you said before, Jim, it's not just efficacy in the animal model. We want to understand where the released drugs are at any given time. That's going to be some of the new data coming for the first of the sustained release. That's going to come a little bit later this month with FAP-exatecan. But really a true deep understanding of where it's being released, how quickly, and whatnot. And so that's the next step with the dual payload technology. And there we're going to have, measures of both payload A and payload B, as we call them, and truly understand, are we seeing synergistic effects in the animals?
Clearly, this is about oncology, but do you see this as being applicable in other fields?
It's an interesting question. FAP, in terms of its expression in other disease settings, the other 2 places I would say that we see FAP expression outside of cancer, one is in wound healing for surgery, and there we wouldn't want to do anything like this. But the one place that we've thought about is the autoimmune field. And is there a place where we have sort of chronic inflammation that could we use this mechanism to damp that down? Very, very early days, not really much that we're doing, but it is something that we've had a couple of conversations about, is potentially, I'm thinking about this in the autoimmune space.
Thinking about it in between all of the other 101 things that you're doing.
You make a great point, because one of the first things that I did as CEO was say, this company needs to focus. And we did divest our diagnostics. We needed to focus on what was successful for us, which is this precision platform. But yeah, sometimes we do think about, could we be doing something in chronic inflammation or autoimmune? It's a great question.
So some excellent news there from Avacta. Lots going on in 2026, shaping up to be a very busy year there, as we hope it is in a positive way for everyone. So that's it for this week's podcast. Time to go and get another cup of tea to soothe the vocal cords and to see if the rain has stopped. If you do want to get in touch with guest opportunities, or if you want to advertise, please feel free to contact me at jim@deeptechdigest.com. So until next time, wherever in the world you are, I hope you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.